Hello, everyone. Welcome to today's webinar titled Current State of PAT Applications for a Reliable and Effective PCM Equipment Operation, hosted by Biopharma Webinars and presented today by Richard Steiner, who is Senior Manager Business Strategy at Pharmatech Associates, a USP company. So hello there, Richard, and welcome. Thank you for joining me.
Everyone.
And we are also joined by Uwe Krishna, who is Managing Director at Centronic GmbH. So Uwe, hello, welcome. And thank you for joining me as well. It's a pleasure to have you again.
Hello, everyone.
So my name is Marcus. I'll be today's moderator. And before we begin, I'd just like to inform our viewers that we will be having a live question and answer session after today's presentations. So if you have any questions for Richard and Uwe, then please feel free to send in as many questions as you like via the questions tab. You can find this button directly below your webinar screen. And then I will ask these questions to our presenters here during our live Q&A session. Okay, and please also state in your questions if they are for Richard or for Uwe or for both of them. That way I can direct the questions effectively. Okay, so now please allow me to introduce our first presenter. So Richard Steiner was educated at the University of Applied Sciences in Nuremberg and the FU University of Berlin, and graduated as a mechanical engineer. He worked in different management positions during his times at Listritz. From 2009, he was heading the Listritz Business Unit Pharma Extrusion. And in 2012, he joined GEA and became 2019 Global Sales Director for the Continuous Processing Technologies at GEA Pharma Systems. And in 2023, he started work for Pharmatech Associates, a USP company, and as a Senior Manager Business Strategy, PCM. He contributed papers and chapters on many international conferences and scientific textbooks, and his favorite topic is the implementation of continuous processing in the pharma industry. Okay, but with that, please allow me to hand over to our first presenter. So welcome, Richard.
Many thanks, Marcus. Many thanks and welcome. Good morning, good afternoon, wherever you are in this world listening to my presentation. I'm very happy to talk about PAT today in the combination of implementation in continuous manufacturing systems. So let me walk you through to a kind of current state of this technology. I had earlier webinars when I talked about unit operations within the continuous manufacturing pharmaceutical process train. And this time I will dedicate my focus on an overview, current state of this PAT technology implemented in most of these systems. So my talk is about quickly control strategy and why PAT is important, sensor technologies along this. Technical solutions. I will focus on content uniformity for a couple of reasons on my presentation. And in the outro, a conclusion to this topic before I hand over to Marcus. Yeah, so we all heard many times about quality by design, the PAT initiative, real-time release. These are just buzzwords which are still around in the industry. And whether you talk to say, yeah, we have achieved all of that, or we are in the middle of it, or some see it even as a hurdle. But I think it's important to just lean back, close our eyes and say, okay, why are we doing all of this? It is, of course, the intrinsic idea to control the quality in a process to not basically create out of spec product. So basically zero failure and always manufacture or process products within a given space of design you have before defined and developed. Whatever the variability of input material is giving you, the process should be able to cover that. And then you have a long time, of course, sensors and parameters which give you an indication how well this process is under control. And then yeah, you can release the product if you work after those principles. PET sensors are, of course, an intrinsic and important element of that. I've just shown here um, control elements of a system company PCM. Some have may heard about this. This is just, I think, a nice example of what typically is going on today. So you have your material attributes inbound. Of course, you have to have material attributes within a specific frame. So the material characterization is important. This hits the feeder, loss in weight feeders is first element of your really control strategy, because these PET or these feeders can be also seen as a PET tool. Then you have a blender, usually a mixing process, where you have then blend uniformity to measure, to control that everything is in balance. Once you go into the compression module, where you have then another area to measure blend uniformity, sometimes in the feed frame. And then on the out, you have intrinsic compression machine sensors giving you an indication how well the process is operating. And then last but not least, but usually still offline, you have then the content uniformity measurement. And I will just talk about this a little bit more down this presentation. So, but you see, such a control strategy is sophisticated, and you have to really know to what purpose you have to install sensors in which element of this, to not compromise the reliability and the operational equipment effectiveness of the whole system. So implementing PET is a process. So you have to select appropriate and representative process analyzers, of course. But the selection of the position where you put these sensors into have to be also very mindful, and is a really important engineering topic. I will elaborate on this. Define the method of calibration, model development and maintenance are other elements to consider. So usually you can start with a mind map. I love mind maps, but this gives you an overview, and you will see there are many technologies, but you have to really identify what is the technology for purpose in your given system. And this is entirely different if you have a powder process, where you have more the physical powder particle structures, or the powder particle characteristics to measure, or if you have a liquid process, or if you have a melt extrusion process. So all these kind of interfaces are important to consider before you even can say, okay, that's the right technology to use for my purpose. Just to mention a few, that you can see how wide today the range of opportunities are. So if I look for sensor-based and non-spectral phenomena, you have sensors from ultrasound, laser diffraction, 3D particle characterizers, light-induced fluorescence spectroscopy. So it's all of kind of really interesting technologies, and I have to feel year by year there's one to add more, and this makes the decision even more complex. But on the other hand, with new technologies, also new opportunities are raising. Here, the typical spectroscopic techniques and measurements, as I said, from radio frequency, microwave, NIR, infrared. We will hear about this, Raman, ultraviolet, X-ray, terahertz. So you name it. So for all these technologies, you can meanwhile find sensors and applications. It's just the point to say, okay, which of my application is important for my control strategy? And you see this on the other side elements of this table. So to decide for that, you have to have an idea what are the critical quality attributes which are impacting my product quality. And then after another screening, so you peel this down step by step, you say, okay, I have this and that control attribute to measure, let's say API unit dose, or API ratio when it comes to blend uniformity. So you look at this and say, okay, what kind of technologies, sensor technologies would fit for this purpose? You will see already on this table that NIR is mentioned many times. And I have to say, even without being a dedicated PET expert over the years, I had also the feeling that this NIR technology has been widely understood, more and more understood, and widely accepted as a kind of the way to go, in particular when it comes to oral solid dosage powder continuous processes. And here just mentioned three of the elements: blend uniformity, moisture content, and content uniformity. Further on, I focus now on content uniformity. What is important is not only that you have a fast and accurate method, but you have to also see what are the disadvantages most likely if you implement this technology to a process. So here's mentioned window fouling, spectral resolution. It is fair to say that in the most of the continuous oral solid dose applications we see today, we talk about new drug products, new drug entities. Also, in many cases, we have to say low dose applications because these new chemical entities are highly potent or unknown. So, in all kind of reasons to say you have a low dose application as well. So, the accuracy and the capability of your system have to be defined to say, can I detect in a certain range? Here, an idea, so low dose applications may be 0.5 to 5% of a weight average, which you see is a little bit the limit of NIR. But I will later let Uwe talk a little bit more about what NIR technology, in his opinion, can come up today. So, but it is important to know at this point already what you are going to do. I mentioned, and this is maybe not always a prominent discussion at this point, so if I have a high potent product, I mean, I have to design a system also under the consideration of containment requirements. Why is this important for the NRs or for the sensor technology? Of course, I have to either position then the sensor in a place where it's automatically contained, but I also can't simply pull this sensor out for cleaning when window fouling happens or something like that, and to stop the entire system by doing that and compromise the containment. So I have to have engineering solutions to make containment as well as a perfect sensor technology and sensor uninterrupted sensor measurements possible. So that's the engineering topic for the process engineers, for the designer of such a unit. Give you an overview. This is a chessboard I've developed over time because I think it's on a high level sophisticated enough. You always have material handling, you have dosing, blending unit operations, you have granulation technologies, whether this is dry, wet, melt, or whatever granulation technology you can think of. You have another dosing, blending rig, tablet compression, coating. These are prominent unit operations. And in the Conti process, they all have to work together. So you have material flow between these unit operations. Unfortunately, or the intermediate material transfer is a challenge in its own right. And so the transfer systems between these unit operations. So it's a mechanical as well as a process interface between these unit operations. And last but not least, the control interface. So you have multiple interfaces between these unit operations. The question, if you are a designer of such a system, if you talk to vendors with these elements, you have to give the question who owns actually these interfaces. Why is this so important for my PT applications? Yeah, because usually you will see that these PET instrumentations, whether this is material identification, blend uniformity, loss of drying, particle size, you name it, they are usually positioned in these intermediate sections, in these interface sections. Not only because it's a logical position, but also because you may have sampling points and diverter points. This all makes it pretty complex operation, and it's fair to say that in the early stages, these interfaces between the system units, you can see one here from a blender to the next one, they are basically unit operation in their own right. If you consider cleaning, CIP cleaning, if you have also level sensors employed. So these systems became horribly cumbersome to manage and to handle. I think nowadays we are much further of this. So there are nice interface solutions designed over the time. But still, you can see on one or the other presentations or installations these kind of complex interfaces. And they kind of give sometimes heartburn in a continuous operation. Yeah. So this is why I say look into your interface functions as much as you look into the selection of your sensor technologies. The size of the sensor technology or the size of the sensor is important if you look into these interface sections to not interfere with these four functions: transfer of the material, supporting the PET sensor, sampling port, as well as discharge for out-of-spec materials. Even ICHQ 13 provided a couple of sentences here: location of material, diversion, sampling points, which means selection of the location for diverter valve sampling ports without disturbance of the material flow and transformation. So it is recognized that that's a challenge, a design challenge to consider. I give you another idea. So powder transfer. You may think, okay, it's not a big thing. But first of all, you have to define if these granules and this powder is wet, dry, or in any kind of way sticky. If you have a gravity, a lean, or dense phase powder transfer. And this gives you a couple of options, whether you put the probe flush with the wall, or if you have to have a probe which interferes with the powder stream. I do not want to talk through to all these elements right now, but you will see if you have a system flush on a wall, you have to make sure that this is wiped off constantly, either by the process equipment itself or by an independent device like a wheel with wiping off, or if the sensor sits in the feed frame, then the feed frame of the compression machine is taking over this kind of activity. But yeah, that's very important to have. Otherwise, you have a more complex system, as you can see here. A nice example from blend uniformity measurement applications. So you can do this within the feed frame. It's a bit hard to see in this little picture, but there's a sensor going straight down to the feed frame. Or you have in the cross section a device like the one here from GEA, Lighthouse Probe, where you have multiple light sources and cross the diameter of the house where the material comes in, you have to measure. It is as well not just the sensor here again, you also have to control the flow of the material alongside the sensor to have a steady and constant flow, to not disturb the reflection of a sensor and your basically data you collect with the sensor. For more advanced solutions today, you will see from vendors highly integrated solutions. Here recently launched a solution from FETI with a dosing blending rig to their compression machines, which includes the dosing blending system, the compression system, which also includes a blend uniformity sensor, as well as a content, or let's better say tablet uniformity sensor, and the outro. Or you have a device like from GEA, the CDC systems. They are also coming with tablet coders and advanced material handling devices. So in these solutions, you can already see from earlier continuous rigs you may have once experienced, either in presentations or in real life. These systems became much more compact, and consequently also the interfaces of the sensors used are much more compact and handy and easier to operate, because all these systems come with a higher OEB as well, containment solution. Digging a little bit into content uniformity as an example, I do not want to read all these guidelines about USP, or European Pharmacopoeia, just to show when you have the job now to say, I have to measure the uniformity of a dosage of a unit, because this is a release criteria in my control strategy. I have to have a device measuring the API content and the mass of a single unit, let's say a tablet. So what are solutions on the market is the big question. And what are the sample size you have to take? Is it enough to have once in a while a sample you measure? Or do you come with 100% check? Question mark. Is it good and necessary? It's a big discussion. And some of the companies implementing this technology are wondering about this. And so what do we see today? There was a device, Bruker Tandem. You can see it on this picture a bit better on the next picture, which was a combination of a typical inline tablet inspection system, weight, hardness, thickness, measurement device, plus a backpack, basically a Bruker MPA transition NIR to measure the content uniformity until to each of the tablets, which have been weighted on the real tablet weight. This was a design of the earlier days, sold a couple of times, but it was taken off the market from the company Bruker because of a couple of issues. It was the limited sample volume, but also the sensitivity and the design of the unit was very sensitive to a commercial operation. Yeah. So data interfaces have been slow and troublesome with all the data volumes. So in short, it was not maintainable to the price of this unit. So many, and if not the most of the installations I know so far out there, went back to the headline testing with Bruker MP as a kind of a standard, for example. So someone is literally taking 10, 20 or whatever tablets, the compendial methods is describing, take them over to the device while the system is running, do the tests in a couple of minutes. It works, but is this elegant as we wished and envisioned to have with a Conti system? Surely not. So company Fette mentioned earlier, they came back with a nice integrated solution. So they have what they call an EPAD, an embedded tablet uniformity NRS sensor just on top of the turret of the compression machine. They can really test 100%. It is a reflection device. So it basically, because it is so fast, the compromise here is you just take the reflection of the tablet surface. You have a blend uniformity measurement device sitting in the feed frame or in the outlet of the dosing blending unit. So of course, you know from all these system parameters that the blend uniformity is given. And then you just say, okay, if this is all right, then the content uniformity, the tablet uniformity is all right as well. To what extent this is accepted is still an open question with your regulators to discuss, and it depends highly on the product, whether you have a high dose product let's say 50 and above percent. So I think it's much easier in this case. But if you go to a low dose application, you might need another system. Here, a company Pharma Technology came up with another nice example. So that's a device with an SRS spectroscope where you not only get deeper into the tablet, but you can cover a wider range of the tablet. While this is fast enough to measure 120,000 tablets. So 100% basically, if you link this up to your compression machine. You have a microwave system for the weight determination or measurement. You have a sizing measurement with a laser. So you basically really have the full capabilities in one unit. It looks like a machine. It is a real independent machine. So, and comes with a certain price. Again, so the question here is whether you need this 100% inspection. I spoke to this company and they say, of course, you can also reduce the sample size. But yeah, to what extent is an open point. Optimal came with a device looking like this. So this basically, if you look to the animation, you will see there's a robot. The robot takes tablets, single tablets to a weighing cell, weighing balance. Then from there to the Puka MPA device. So that's nice, fully automated, but also equipment in its own. There's grammar offers as well a transmission NIR spectroscope, which after the certain regulation takes a couple of samples and is not full 100%, but a nice integrated system with the typical tablet inspection devices. I did a small overview here. Yeah, you see what you have on the market today, and some are faster, like the ePad. Pharma Technology a little bit slower. And here I want to again go back to the containment point I have brought up. So it's worthwhile looking to this because you have to integrate these devices maybe in your commercial system. So they have to have capabilities of containment. And back to the point, most of the compounds I have seen on those devices are in the range of OEB4, if not 5. Yeah, so these auxiliary equipment providers, they have to work on additional containment solutions to make this really applicable. Let's talk about other critical process parameters you have already on board, not as a sensor, but embedded in your process technology. If you go down this process train, the theoretical process train, you see, of course, you have blender speed, you have loss and weight load cells on either the blenders as well as on the feeders, which allow you to see immediately if, for example, bulk density is changing from your inbound material. So these loss and weight feeders, they are sensitive enough to give you ideas and signals if there is some change of some of the material characteristics already before the whole process hits the other unit operation. You have screw speed, torque, so you have a lot of process parameters already on your system, which will give you an indication if something is changing outside what you have defined the system for. So your quality by design, your design and your testing before to define the design space and the parameter space is crucial and helpful because you can run such a system basically without an online sensor once you are sure which of these critical process parameters are showing what effect. Yeah? So also for the compression here, you have force, displacement, speed. So you have variables which indicate you if something is changing. Putting on all of that multiple PET sensors will create, of course, a huge data stream, a huge data volume, and additional complexity, which is very well handled with PET management systems today available. But again, it's complexity, it's costs involved, And you have to really be sure that you implement PET analyzers for a specific reason. In the sake of time, I also want to say what is more and more common and understood and accepted is the use of process models, so predictive models. Predictive models also per ICH Q13 are possible to be applied, developed and implemented to be part of the control strategy of your commercial production. So let's say you develop in your R&D space a predictive model for your Conti system, then you can run most likely the whole system without sensors on board or just using the sensors as monitors of your process. There's nice studies out there you can find, for example, this one here, 120-hour run with a couple of well-known people in the industry confirming this, where you had a content uniformity measurement, basically at line. The whole system was operating around 120 hours with predictive content uniformity models, very well aligned. Whenever they took samples with the at-line system, it was spot on. So a nice proof. And so in this control strategy, the content uniformity was an at-line measure. Also, if we go to the ICH guidelines looking into real-time release applications, the same thing here. Yeah. So it is mentioned in these guidelines that you can use predictive models as a form of parametric release function. And then you do at-line just checks, which you anyways have to do before you release the product. And I think, and this is one of my outros here, implementing this kind of sensor technologies, which gives you, of course, enormous amount of data and understanding of your process, is a good thing in the development phase. But you also have to go back to the idea originally of continuous manufacturing. So why you implement continuous manufacturing in the first place? And the simple reason is because it is evident that this is a Six Sigma process. So you use this technology for a purpose, that you have a much more reliable process, which you can prove with process capability factors. You also can develop and calculate for your process to see that you have a Six Sigma process and you have designed your equipment well, that this is a fact. You can show proof of that. So then the question is, if I have this kind of Six Sigma principles applied and I have an operation like this, I have little effects to expect. Do I really need 100% check for that? Companies like ours are in the field to give consultancy about these kind of questions. We use then tools like the OC, the operating characteristics curves, with our statisticians to develop an answer to this question: what is the necessary sample size for a process like that? Which is, of course, necessary to develop case by case, as it is highly dependent on the product itself, not just the process equipment. So, but you can find answers, and you will then may see that there's no need for 100% check. And to everyone out there, we have continuous operations, we have continuous equipment, and today the experience is that the reliability of a PET implementation has a big impact on the performance of the equipment, but also the yield in a positive way. So you hear a lot of arguments that, oh, with PET I can improve the yield, but you hear little about the input on the performance of these systems. And the business case for PET is about exactly this balance between these two factors. To give you a little idea as my outro of this presentation, so if you go into a business case calculation, you calculate your net present value. Let's say this is your batch system. It will operate on an AE 50%, just to say something. Over certain batch sizes, your net present value might look like the orange line. If you do the same with a Conti system and operate this Conti system on the same OEE level 50%, you see that the net present value of the batch is still better than for the Conti system. You may wonder, you say, hey, I thought it's better. The simple difference here is if you have not produced more output on a Conti system, you have the disadvantage of the higher investment costs. And these higher investment costs are even higher with implementing PET on your system. If you would be possible to increase with your PET technology the efficiency of the system, so the yield, and you would then go into a higher yield operation above 50%, then you can, of course, balance out the negative impact of your investment, of your higher investment. So the question is, how high can I go with my OEE? And this is where the performance of these sensors are playing a big role into. So you should never compromise with the implementation of a PT sensor, the reliability of your operation with this kind of unit operations. Yeah, and recently, and this is my last slide for everyone, recently also a speaker on one of our conferences mentioned this, saying, okay, in our case, this is an irony. We found after many years operating this that our systems are really Six Sigma capable. So we could theoretically switch off all the sensors and we still Six Sigma operating with our product, with our given portfolio and our equipment. So, but we have now the problem that we have put all of this complexity in our product filing, in this control strategy for our product filing. We can't wheel back anymore, unfortunately. Which days for whatever comes next for this particular case, they will have an eye on in the earliest selection of the reliability and the complexity of the system. And yeah, with this, many thanks for your attention. I hope I was not eating up too much time of my successor now, of Uwe. Sorry for that, Uwe. But I think it was important to note that reliability is a factor in today's PET environment. Thank you, everyone, for listening to my presentation.
Okay, thank you very much for that, Richard. Great presentation to start us off today. And to our audience members, if you have any questions for Richard, then please do continue sending in your questions via the questions tab found directly below the webinar screen. And we will ask Richard and Uwe your questions later during our live Q&A session. Okay, so once again, thank you, Richard. And now please allow me to introduce our next presenter. So Uwe Kirschner holds a degree in electrical engineering from the Technical University of Dresden. And already during the last months of his degree course, he founded Centronic, where he spent his entire work life so far. Together with Matthias Lau, he started to work on the development of sensors based on optical technologies. At this time, Centronic also established itself as a distributor for spectroscopic components. And in 2000, the knowledge gathered on the spectroscopic components was used to start the development of process systems based on optical spectroscopy. And from the beginning and still today, Uwe is the head of the business unit process spectroscopy at Centronic. And in this position, with the strong focus of PAT for the pharmaceutical industry, Uwe gained knowledge and experience in many aspects, in particular in instrument development, NIR spectroscopy, PAT, chemometrics, GMP, and several others. Okay, so now with that, please let me to hand over to our next presenter, Uwe, and we'll set up your share screen now. Okay.
Thank you, Markus, for the introduction and for the invitation as well to Richard for the first part of the talk. Thanks a lot. I'll try to dive a bit deeper now into, of course, a more specific topic, compared to the broadband presentation Richard gave on the topic. So I'm going to talk about the NIR PAT applications in pharmaceutical continuous manufacturing applications. We are focusing on oral solid dosage forms mainly. So just a brief intro. We are running Centronic since 1993. We have our main offices here in Germany and Dresden. We have beautiful weather today for one of the first days here. And we have a subsidiary in the US run by Owen Rayrauer. He is in Norwalk, US. We are focusing on two major areas. So one is NIR PAT applications and solutions, with a majority of our, say, experience in the pharmaceutical industry, where we started working around 2003. And we have a second product line in that business unit as well, which is handheld IDs, mainly used in the German pharmacy market. There are many units out there, and we started that business around 2013. We have a business unit for optical chemical sensors and solutions, dissolved oxygen sensors, mainly for aquaculture applications, which is run by my partner, Matthias Lau. And I'm going to focusing on the PAT applications in the talk. So, a short overview of what I want to talk. I want to give you a slight overview where we are seeing NIR PAT in pharmaceutical continuous manufacturing, again, just for oral solid dosage forms. Then I will introduce you a bit to the solutions for blend uniformity measurements in continuous manufacturing and the different measurement positions which are available here or which are possible to do BU measurements. Then I give you, and we'll have some examples from this area. Then I want to show you some results from tests just done for measurement at low dose in a feed frame simulator, and a very quick outlook in the end. A slide which many of you have seen before is just our overview page with the product lines we have. So we started to work in the pharmaceutical industry, as I said, 2003, and the first really dedicated product in this area was the Centrapad BU, which you see on the right side here. It's a blend uniformity analyzer for container blending, which was and is still an interesting application and an important application for batch operation. There is a new product available from us since about 2022, which is a diode array-based smaller version of the BU analyzer, fitting the same requirements and for the same applications. And then in around 2010, we launched the Centrapad FO, which is still today the most used instrument from our product line in that area. And the applications we are covering, they have been starting with granulation and drying and blending in the beginning, compression, encapsulation. We have really done quite some things on all these areas, but the majority of our work is really related to blend uniformity. On the batch side with the Centrapad BU and Centrapad DA, and on the left side with the Centrapad FO, mainly for continuous manufacturing operations. So that's why the rest of the talk will mainly focus on the applications of the Centrapad FO in continuous manufacturing. So we'll focus mainly on the continuous direct compression applications. They are very, say, widely used meanwhile in the industry, and they get the most of the attention in the industry. I would not say there's still a lot of granulation and other high shear granulation and also roller compaction applications. But most of our work is really based at the moment on the continuous direct compression application. And there we have 3 different positions where we can actually measure the blend uniformity. First is not only accessible, but there are cases directly at the blender or in the blender. Secondly, somewhere between the final blender and the tablet press, quite a common place as well. And then finally, BU in the feed frame of the tablet press. So these are the main positions where the instruments can be used in a CDC line. Starting with one of the examples, here we see a Garrigue line using a TESO interface for collecting material, and then a spoon is just flipping every few seconds to replace the sample by a new sample. Actually, this development was mainly for spray drying and other low volume applications, so it's dedicated for really low flow rates and low product flow rates. There is a spider wheel alternative in development, which will then be more suitable and collect more of the product material in the same time by a continuous rotation. But okay, this is about to come. A measurement directly at the blender is possible, for example, at the Garrigue mini blender version, where the blending process takes place inside this blender tube, so to say, and probes have a central probe has been used there already. Some slides about that will follow a bit later in the presentation. Then the next position between the blender and the tablet press is quite common in the GEA Consigma lines and is based on the central part FO with the central probe MS7. On the upper side, you see a view from top into the powder section, where the powder is slowly flowing along the probe with the 7 spots. These 7 spots are just elimination spots from a fiber optic probe, which is just 12 millimeter in diameter. The probe can be moved in different positions, from the measurement position to a reference position, and optionally also a washing position. Here we have a very stable powder presentation controlled by level sensors usually, and have a very, say, constant and defined product number measured. There's also a little more slides coming about that topic later on. Then measurements on the feed frame. Usually a probe in the feed side can be in place, can be installed from the top. The probe goes into the process usually without an additional window, is mounted, fixed, and is established in many different press models in the meantime. The measurement here provides a possible input for CU, and you can measure the blend potency here and the blend uniformity. Actually, the measurement in the feed frame is a very good location because it's the last processing step before compression. The powder presentation is really very stable. Fouling issues on process windows are not really very likely here, because otherwise the material would also not be that well processable in the tablet press. Another example, after the course, is also available in the Fette press and Courtois, of course, as well. This is how typical installations look like. So what use cases do we see here? Yes, NIR spectroscopy, as you may know, is very information rich. So it contains information about the chemical composition. It contains a lot of information about physical properties like density, like particle size distribution, are all affecting NIR spectra, which is good and bad on both sides. At least it gives you an eye into the process, which is more than just a sensor or software model in that case. So whatever happens, the NIR may respond to whatever type of variation. So it's useful in this case to support process understanding and process characterization. It can help to speed up the development process in a continuous line. It's also supporting the DOE development or the residence time distribution models, because of the API concentration can be measured in real time or trended in real time. And so RTD models can be either developed or verified. During manufacturing and release, there is process monitoring always ready as a steady state control of the process, assuring that everything is really in line. And also it is an enabler for real-time release based on blending PAT and tablet PAT in combination. And in general, it's a great tool for process transfers as well as for troubleshooting. An important factor, usually lines should not be or would not be, say, changed after they got installed. So installing PAT from the beginning allows you to do later on product developments on the same line when you might require PAT. So of course, I'm talking for the PAT here. That's our company's business actually. So, but on the other side, we wouldn't really think that PAT needs to be installed everywhere and for every reason. So please consider where to use it and why. Now I want to give you a quick overview on our systems for those who do not know them. Well, it's not much technical, it's just an overview. So here we have the single channel central part FO. This is actually the most used instrument in that type of applications. It's diode array spectrometer with a 1100 to 2100 nanometer spectral range. It has an internal built-in Windows computer. Actually, at the time when we developed these instruments, it was 2004, 5, and 6. Having a Windows computer on the system was really very beneficial because we were able to qualify that systems in factory. And there was not much difference between qualifying them at Dresden or qualifying that at the customer side. Meanwhile, it's not that big advantage because of increasing IT security questions and issues. So a bit more containment sometimes is good, but okay, for now, this is what we have and it still is well. Fiber optic is connected here. Then let's go over to something weird. Okay, let's go have a look into the central probe, the RLS. When you take a look into the tip, you see two rods here. These are the two light sources. One is just illuminated, the other is not. Then you see here the internal reference, which is one of the most important features of the systems. It flips on a time-controlled base, measures an internal white reference, and goes back to measurement position. And so it allows to correct the complete system for long-term or drift effects from temperature or from long-term changes over lifetime of the probe and for very long periods. So the window can be heated and temperature controlled, which is beneficial in a couple of processes where raised temperature at the window reduces the risk of window fouling. The system has two built-in lamps, so it's possible to activate a second lamp without operator changing lamps on the system. The lamps are actually interchangeable. And these probes are ATEX certified for dust zone 20, zone 21, which is a requirement for quite a couple of these installations. Inside the central part FO, we have an internal wavelength standard, like a Fabry-Perot type filter. And whenever you see a wavelength shift happening, the spectrometer will be corrected back to its correct original wavelength axis. So it's actually locking the x-axis. There are versions of the instrument which are temperature controlled. So that means the whole instrument itself can be temperature controlled, including the box. This version you see here now is a version with a 9 watt internal light source, which is used for the MS7 probes. The MS7 probe is a probe with fiber optic illumination. Different probe length versions are available for different applications. Sometimes there's not much room for a long shaft. Sometimes the long shaft is needed. So they range from 65 millimeter to 300 millimeter. And also shown here is a 4-channel system. This is the alternative to the single-channel system. It's not as widely used in the industry right now, because in many cases where PAT is used, it should be there continuously on a single measurement channel, even though the multiplexer is fast. On the other side, also the risk is seen higher if a system with 4 channels would fail. All 4 measurement positions would fail. If a system with a single channel would fail, it could be easily or better replaced by a redundant system. Okay, let's come to a very important question. That's instrument standardization. So what we all would like to have is, if 2 spectrometers are measuring the same sample, the NIR spectra measured should be identical. I mean, we all know this is technically not possible, but we want to really come as close as possible to that point. So when 2017 Pfizer was developing on the PAT systems for the PCM lines, we had a workshop in Dresden where 6 central part FO units were used to check also their performance and also transferability. Actually, Steve Hammond was doing that, but Steve is actually behind the camera, so that's why you can't see him. This was actually also the first time the benchtop feed frame was used for the purpose of these trials. And so it's an early version of what is being used later on in the industry. And also you haven't seen Sebastian, but this is just Sebastian's coffee cup. When we started that work, the instruments we looked at had a quite large variation between each other. So we had testing them with, they were tested with an APAP blend, and there was quite some instrument relation to that. So we had to do our homework to see what's wrong and how can we improve that. And so we developed a new adjustment method for the x-axis, which was a major improvement. And after that, the x-axis, or say the responses in the methods, have been much, much closer to each other. And so what did we do? We have been calibrating or adjusting our instruments until 2017 based on the certified peak locations, for example, of a 1920 wavelength standard. This is just the green mark points here. We were then changing that method to use peaks from a second derivative spectrum from the same standards. And so from 6 peaks, we got to more than close to 65 to 70 peaks usable for the x-axis adjustment with a very nice spread over the complete range. We were also using several NIR wavelength standards for the master calibration we built at this time. And since then, we are calibrating our instruments with that method. So, and what you can see on the right side is this is the standard deviation for all FAT and IQOQs we had performed between 2018 and 2023 on our systems. And we looked at the peak deviations and looked at the standard deviation of these peak deviations. So that means on average, the peak deviations of all our instruments is within, say, 0.2 nanometer standard deviation, not max to max. But you know, the USP and EP requirements are something like plus minus 1 nanometer. But we know from experience that plus minus 1 nanometer would be critical because we had always maintained that plus minus 1 nanometer, but still the instruments had more variation than they should have for seamless method transfer. Okay, there is a derived publication which is just a bit based on that work from the same group actually, or from people at Pfizer. They were using multiple NIR instruments and built a method just on one of these instruments, on one development unit with a benchtop feed frame. This is actually a published work you can see. It's actually using a similar feed frame simulator as used in the trials in Dresden, and one dedicated central part FO instrument. A full DOE was performed on these to build a method for saccharine and to test this method then on other instruments on site. So this method built on the single unit with the mini feed frame. The method was built with a low amount of material, so it was just very little material required for that test. And this method was then applied to 2 manufacturing sites, PCM manufacturing sites, with having their own NIR systems. There was no method adjustment done during this transfer, and this method has just been used as is on the 2 instruments. And then validation plot is showing a very narrow and very tight calibration, which is an acceptable accuracy for a potency prediction for these 2 sites. Actually, the actual concentration of saccharine was around 20% for this trial. Another example, similar topic, was the question of to analyze the lead lag, so the time and volume between material being in the feed frame until it's going into the tablets, investigated by NIR systems as well. So in this case, it was University of Ghent and Pfizer in cooperation, and 3 systems were used in parallel, 2 in the feed frame of the tablet press in the 2 different sections, and 1 in the hopper. The measurement in the hopper was a bit slow, of course, because material flow is really slow here and was not that relevant for the outcome of the of this paper, but it was an interesting observation how these 2 NIR are predicting related to the tablet content uniformity after the tablet press. The DOE was studying a couple of effects like pedal speed, turret speed, and formulation properties. The overfill did not have a much effect. So here are some example trends from the same publication showing the results of 2 online NIRs and the content uniformity NIR, the tablet NIR in the end. So black is the first in the feed frame. Second one is the feed before tableting, and the green one is then the confirmation by the tablets. So you can see slightly different profiles here. You see the fast response of both, and actually this was a step change which was then tested. Here you can also see that this can be used also for a diversion strategy. So that means if there is a spike, for example, in API high or API low alarm, the feed frame NIR is responding really fast and detects the upper control limit quite fast. You can see on the second NIR in the tablet press that it dilutes out a bit slower. And you can see on the tablet NIR that they also respond a bit slower, similar in the line as the second NIR from the tablet press. So this means that the NIR here can create an extra layer of confidence about the final tablet quality. And it also can be used to potentially control a diverter to improve the yield of the process. Short overview on another, say, possible setup for continuous line is the Gericke mini blender. This is how it looks. This is a chamber in which the blending takes place. It is being filled, blended, exit, or emptied, and again, it can be integrated in the continuous lines with a cycle of process between feeding, mixing, and discharge. This is about the process itself. Though you have the powder or the blend in the blender, you have the blender principle here. And yeah, it is PAT ready, this process. So PAT can be installed here as well. And similar to other applications, you can continuously monitor the blend process, and you can also get information about the blend uniformity within that process and the blend process itself. This, for example, is a step change, is the addition, is a run with a 10% paracetamol added. And you can here see that's before the blend curve, before the blending starts, and this is during the blend and the continue or the stable phase of the blend. Now, one example from Central Part FO with the Central Probe MS7, which is the Lighthouse probe. This is a publication which also Richard already referred to. Richard referred to that from the content uniformity side. I'm referring to that for the blend uniformity side. Actually, this is about the installation. Apologize for the quality of the picture. It's not great. It was from the publication, but this is actually the central probe itself. This is just a pure probe with the optical section in the middle by the round sapphire window. Here it is integrated in the LHP housing. And on the left side, you see the Lighthouse probe being installed here in the middle. And the whole section here is a controlled powder section where the powder is controlled in this hopper by the level sensor. So the level sensor and the feed or the wheel above are controlling then the amount of powder. In the probe is also a calibration. So the white reference is being done by retracting the probe from the process automatically, placing it in the white reference position, taking a new white reference and pulling it back into the process, which can be done as necessary. Usually it's time of hours between those process steps, between those referencing steps. Here an example plot from this publication. It's actually the blend potency for a couple of batches monitored over time. So this is just an example that the blend potency here is not part of the control strategy. But it's used for RTD method development or model development and process monitoring. Here also the NIR supports the development and allows the monitoring of routine operation of normal operating batches. Now a larger section or a section about trials we have done with feed frame simulator and low dose formulations. So actually we were interested to look a bit closer to the limits of the quantification of NIR methods. So I was going to Ireland to do trials with Phil. It just was the beginning of the year, and one of the outcomes was a presentation at IFPEC I was giving last week, and so we wanted to really look at what can we measure with our NIRs in the feed frame simulator. We did the mixing in the feed frame simulator itself. That means we did not pre-mix anything. We just were using pure components in the beginning. We're weighing them in very precisely. And then we were just loading them into the feed frame and using the feed frame simulator as a blender in this case as well. We'd see data on that. Then we did two things. We had MCC as the main material, and we added first APAP to up to 10%. And then also we did a magnesium stearate addition with up to 2% in the end. Data is being followed. Phil and me setting the whole thing up. And here now we can see actually the filling process of the feed frame simulator. So we actually just starting it and filling it from the left side here with, I think it was something like 80 something gram of MCC. And yeah, I told you before that the diode array spectrometer is very fast. So what we did is during that trial, we were monitoring the baseline, or we monitored all spectra already during the filling process. On the right side, you can see that we have done these trials with two Centropad FO systems and their probes. One is a 300 millimeter, one is a 200 millimeter probe. All the time they were measuring simultaneously in the process, which also tells us, hey, we can either do model development on two units in parallel, or we can do, it is also possible to use two different probes on the feed frame simulator to either compare different technologies like NIR and Raman, or two different instruments or sensors, or like in our case, to just also look at transferability of methods, etc. Yes, now let's look at something we are using for an outlier rejection. The blue curve on the right you can see is actually only the absorbance value of the spectrometer or of the spectra at the wavelength 1300 nanometer. 1300 nanometer is a wavelength where nothing else is going on. And so this wavelength is good baseline. And you can see before actually the process of filling is started, we have a stable baseline with a high value. It's just a background signal within the feed frame simulator. Then we are starting to fill it, and we can see the curvature of absorbance values going up and down. This is a good example also how the outlier algorithm is working. A single measurement is really very fast. Every blue dot is a single spectrum taken. The rejection algorithm just rejects everything which has a too high baseline, because high baseline means bad sample presentation. In this case, it's just usually the distance of the powder bed between the window of the probe and the surface of the powder, which in our case was not constantly connected to powder. But with this outlier rejection algorithm, we can remove all the non-well presented spectra quite efficiently. So what we then do is, depending on the average settings, here in this case it was 60, we are taking 60 spectra and we take all the good spectra from within these 60 seconds and generate an average spectrum of the good spectra for that time. This is what we actually then use for further processing. Okay, what's the effect of the outlier if I use it or if I don't use it? Here you can see two different, two instruments. One of the two in this trial had outlier rejection turned off, while the other unit had the outlier rejection turned on for one process test only. You can see the unit with the outlier rejection turned on provides a really stable and really low baseline. While the instrument with outlier rejection not turned on has a much higher variation in absorbance values in baseline. Okay, but what does it mean? We can actually do SNV correction. So we do a standard normal variate, and after that, well, all these data which have been so different now look extremely similar. So most of the variation is gone, which actually means that it's correctable for this distance to the most amount. But let's take a look into a region like here, this red marked area. How does it look here? So here you can see that even though they are very similar now, still the variation is a bit higher when we have the outlier correction turned off instead of turned on. Of course, it's all a question of sample presentation. So if your sample presentation is really very good and you have no variation like that, then that wouldn't really create a large effect. But especially on dynamic situations like in the feed frame or in other locations in the blender, after a blender, you would have this variation. This variation is less on the MS7 version, where the powder flow is very much controlled. But let's see, with the outlier, what can we achieve with this data? So all the data I'm presenting now is just taken in the feed frame during the trials. We were not using challenging materials, and we can see we used MCC, paracetamol, and magnesium stearate, which are quite well. I mean, they are also used very often in similar trials and studies because they are not very harmful and they are good for these kind of operations. So with our outlier rejection in place, this is how the absorbance spectra of the mixing of APAP into MCC are actually looking. This is just pure absorbance spectra. No pre-processing was used for this data. Usual first derivative SNV really pronounces or say increases the variation between the different steps as well. Okay, and these are the data which we're then putting into a method. And we challenge this developed method with a very low concentration of APAP into MCC. The calibration itself was built on a large range from 0 to 10%. And then on day 2, we were just challenging the instruments with stepwise increments of 0.1% APAP up to total 0.5% in total. So actually we're looking at how can we deal with low dose. Each concentration level was operated for about 5 minutes. The PLS model used had 2 factors because it was using instrument from both data from both instruments. So at least one factor was also required to compensate for the variation between these instruments in a way. Owen was preparing me this nice prediction distribution slide before, and it clearly shows that at very low concentrations we have a separation, a definite classification of 0.2%, 0.1% more, and on the higher side we are closer to 0.2%. It looks logical, or at least it's a possible root cause that the remaining inhomogeneity of the material measured in the 1 second spectra, because all these spectra were just taken for 1 second, which is not a high sample mass measured at this time. So we are not sure yet if this is the end of story or not on this application with further pre-processing. A second trial was about MCC and APAP blend. We had a 0.1 step and then another 0.51 to 2% mixing of APAP into MCC. And here we see the second model. It is using a 3-factor model in this case. First derivative, SNV, pretreatment, and 3 factors used. We did this analysis using the vector director, which some of you may know. Then we tested the magnesium stearate addition. Actually, I must correct. This is magnesium stearate in MCC, in the blend we did before. And these are the data from both instruments which were used to create also the method. You see here also the blending effects on these. So every step here is just the addition of magnesium stearate into the blend, and you can actually see the blend curve in the feed frame and some spikes like lumps in the beginning before the blending is really done. And you can also see that both instruments are behaving very closely, very similar to each other. So they both see actually the same spikes when you look at those. More or less, both instruments have been just say 120 degree away from each other on the feed frame. Yeah, that was what I wanted to show you about these trials we were running. And now let's shortly conclude the talk with what are our future plans for our product development? Yes, we see the need in the industry moving to lower dose products. We want to do the best to make sure that the instruments can really perform as good as possible on low dose products. We'll keep looking into that. We keep our focus for NIR and OSD manufacturing and pharma. So that's what we did over the last years, and we have no plans to change that. Then we think it is good to work on all-in-one systems connected via OPC UA and no longer running, that means no longer, having alternatives to our current Windows-based systems sometime in the foreseeable future. And also we want to simplify operation and instrument control, which we think is very mandatory as well. Richard mentioned the cost of PAT. Of course, the cost of using PAT is also dependent on reliability and ease of use and maintenance questions. So we understand this as a hot topic, and that's why we really focus on that. And on this road to this development, some of you may have seen this before. We have been working on the new product, the Central Pad Compact, for quite some while now. We have shown it at IFPAC the first time in operation to our visitors at our booth last week in Bethesda. We had nice conversations about that and interesting talks. This is how it compares to the Central Probe, the RLS. So you see the housing is slightly bigger than the DLS, but it comes with the same length options for the tube. You see only one cable. Then here is the fiber optic and the electrical cable, and here's just the Ethernet cable coming from that unit. This is just a direct comparison to the Central Pad FO and the Central Pad Compact. You see it's, of course, quite compact system and still contains the functions of the Central Pad FO. So that has about the same core functionality. Just the internal embedded computer is smaller. It's running on using Linux operating system, and spectral range is 1100 to 1900 nanometer with a decent 10 nanometer full width half minimum spectral resolution, which from our experience is quite a good spectral resolution for the requirements of resolving as much as possible from the API concentration in a blend. It's just connected by power over Ethernet by this one connector. And the lamps are, or the system is actually maintenance free. These lamps are supposed to be lifetime lamps. That's the goal. That's what we think we can achieve. And still these lamps are replaceable. And again, it's two lamps, same as in the Central Probe, the RLS. So even in case one lamp would fail over time, there would be the chance to just switch over to the second lamp and continue operation with that. Yeah, we are in the final phase of development, working on topics like ATEX certification, series production, preparation, slight redesigns after the first prototypes, evaluating the equipment, and we think that by the end of the year or early next year, we should be able to deliver the first instruments to the market. Yeah, that's about it. Thank you very much, Owen, and thank you very much, Sebastian, especially to you as well, because I kept you busy all this morning, because I was again quite late with my presentation for the talk because there's so much to do in advance. And thanks to everyone who responded to my last minute questions. I think you, the ones who answered, know who I mean. So thank you very much for that really last minute answers you gave me to some of the questions I had while I was preparing that presentation. Then just a final word in our own behalf and interest is we will have our next PAT meeting in Dresden in December. Okay, now we have nearly spring and we're looking for spring and summer first, but yeah, the year will end at some point, and short before that, we'll invite you to join our PAT meeting in Dresden. And if you are interested to present, just contact us. You probably know how to find us. Yeah, thank you. That was all I want to say around that. Giving back to Markus.
Okay, great. Thank you very much, Uwe, for your presentation there. And thank you as well to Richard for his presentation beforehand as well. Much appreciated. It's always great having you here. So in a minute, we're going to take some questions. I'm going to ask them to Uwe and Richard that we have here. And if you have any more questions while we're asking them, then please don't hesitate. Send in as many as you like via the questions tab found directly below your webinar screen. Okay, so once again, thank you, Uwe and Richard. And our first question today, I'll ask to Richard. And this question, Richard, here says, what is your experience in regards of the usage of PAT in OSD continuous manufacturing systems? What sensor technology is used most and why?
Yeah, I think Uwe gave a nice answer to this question in his talk already. I think it's pretty obvious that NIR technology is the one which is used in almost all of these applications. I know about it. And then you look closer to it, it's usually blend uniformity applications. There's not so many in the granulation section, but yeah, it's all about blend uniformity after the dry blend and the blending device. There's maybe one other technology, is the particle size distribution measurement, which is also kind of seen here and there. And then, yeah, lastly, you can have content uniformity offline. Yeah, so these are the three. I forgot material identification. I think that's also, but here you see the range is always about NIR and specific this technology, because it's a reliable technology. Meanwhile, and Uwe showed it very nicely how advanced already the design of these sensors is going to be much more compact. They are not looking like lab development and playgrounds. They are really solid designs now available in industrial standards. That's great to see, Uwe. Compliments.
Thank you very much, Richard. So before our next question, very quickly, Uwe, did you have any additional comments to follow from Richard?
Maybe just to mention Raman as an additional technique, not as widely used in these applications yet, but of course trying all the best as well to get into that market segment with partially really fast measurements in the, say, 50 milliseconds, 30 milliseconds, 80 milliseconds integration time. So that will probably be, yeah, they're more specific, but still sample position, sample materials, sample volume is even lower than with NIR. So there are questions around that as well.
Okay, great. Thank you for those answers then. So our next question to Uwe now says, there was no specification on ATEX rating of Centropat Compact. Will that system be available in a dust ignition proof version as well?
Actually, it's very similar to the central probe, the RLS, and we will plan to go for the same ATEX certification route as with the probe done before. So, but it still has to be done. So it's one of the question marks on the timeline, I would say.
Yep. Okay, great. Thank you, Uwe. And very quickly, before our next question, Richard, do you have any additional comments, thoughts to go with Uwe's answer?
I mean, I highlighted the containment part, and if you will, this also goes into this ATEX scenario. So it's all about cleaning, it's about accessibility of the probes, it's about the more industrial discussion now to use these probes in an industrial setting. Where it was 10 years ago, it was more in an experimental level. Yeah? So you see very nicely on this kind of discussion that this technology goes more and more into commercial use. And I'm glad for this, and I'm happy about these things. This is what the process engineers and the engineers on site want to know if you sell equipment. Yeah? Say, hey, what is about this? What is the capability? Where are the risks employed? And what are the benefits of it? So I think we are much better prepared today to answer this.
Great. Okay. Thank you, Richard and Uwe, for those comments there and answers. This next question to Richard says, what is the effort to develop full skills in PAT, and why is it important for any implementation team to have those skills on board?
Yeah, I think I see in particular companies who are interested in continuous manufacturing but do not have these skills or the skill set among their team. They are struggling with the first steps in a lot of cases. So they come to us with very basic questions, with basic training requirements, and say, hey, what can we do with that? Yeah. So what is this? Really the basics. And so I think you need to have in these teams on the customer side, you have to have really skilled personnel available, which have the capability to understand the complexity. So it's not just a plug-in, it's not just something easy and simple. You have to know about chemometrics, you have to know about the physics around these instruments. And that's a skill set which is really needed in these teams. Of course, you have vendors which can partially fill into that. But then if you have to operate this equipment, I mean, I showed you the availability and then the performance factor. It is all about the team you have on board as well, partially for the initial section. It's an R&D and a development skill, but then you also have to have people to have the understanding. I mean, simple comparison. If you sit in your car and the tire pressure sensor gives you a signal, you have to understand it, have to make the interpretation if this is now a sensor who is just giving you some advice, or if this is something what comes up on your screen which is really relevant to interact immediately in your process. So you have to have these engineering skills and understanding on board. This is essential, I find.
Okay, thank you very much, Richard. And Uwe, do you have any additional comments to go over this?
Maybe just one, which I did not have in the presentation as much, that also there are PAT management solutions on the market, which incorporate many different PAT systems into a same software platform, like SciPad, like SyntiQ, like PharmaMV. They take a lot of the burden of integration if they are going to be used. And there is some room for improvements with that interfaces so that they can be used for more or less everything. Except maybe the vendor work when we are doing qualifications. But this is only part of it. Besides that, Richard, I agree with you. It's a very complex technique. It requires interdisciplinary teams to work on. But there is also a lot vendors can support that process by, say, trainings. There are really many experts out there who are helping customers in implementing PAT for many years with background and experience from the industry. I don't think I need to name them by name, but they are really a kind of active group, and they are supporting also customers who don't have that much internal resources. But
come to the next IFPAC. You meet them there.
Yeah.
Okay, great. All right, then. Thank you very much for those comments and answers. This next question to Uwe now says, the accuracy of predictions for the low-dose experiments looked really impressive. Can we expect that all of those ingredients we used can be measured in such low ranges and with such accuracy?
No. It's a sugar and salt question, so I would say. You could predict sugar very well. You would not be able to predict salt if it has no NIR spectrum. So a lot is depending on the NIR response of the substances. So to take a look at what your blend is composed of, and really make sure that you see that API has enough difference from the excipients used, for example, that the wavelengths are not overlapping and strong enough. So that there will be limits where the predictions or where the calibration models will not be as accurate.
Okay, thank you, Uwe. And Richard, any additional comments here?
Yeah, I just can fully highlight what Uwe was saying. I think that's I saw with my own eyes products which have been really almost invisible towards the spectra from the excipients, and then it's a kind of, oh, can we use this technology? And you have to find maybe other solutions than in this case. But I wanted to show the overview of all the existing technologies today to give everyone an idea that if it not works with one solution, have a greater look to other solutions. Don't give up and say, yeah, there will be something to discover. I also would want to always highlight the need of looking into the position of your sensor very carefully and to make sure that he sees everything possible. And I think this is also crucial in the layout to consider.
Okay, great. Thank you very much, Richard, for those comments there. So this next question to Richard says, what other sensor technologies was, in your point of view, successfully implemented so far?
Yeah, I think Uwe gave already an indication, Raman. So I can recall a nice application where Raman has been used for tablet coding solution. So it's really a great inline measurement device if you have space for it on your device and you have a dedicated area where you measure it. And also if the reflection is meaningful for the product you are measuring. Yeah. But Raman, I think, has also great potential and shown great solutions, very reliable, also for a longer operation reliable.
Mm-hmm. Okay, thank you, Richard. And Uwe, any additional comments here before our next question?
Just because Richard mentioned the coating process, he had it already in his slides. The OCT technology is also interesting for coating and for the physical properties of the tablets being measured after coating. So
one point I forgot totally in my presentation. So you have to see also there are processes where it's really required to have the sensor technology on board to make sure that, let's say, a reaction process is fully finished or things like that. So where you really directly need the sensor to determine if the next step of the intermediate has been happened. So you need these direct measurement sensors, whereas there are other sensors where you more or less monitor the process. And if you go more to the API manufacturing space, you will find much more implementation of PET for really getting the point of the process done before you go into the next unit operation. So I think there's some distinction necessary to make And yeah, in PET, never you can generalize anything. You have to really dig very deep into the weeds.
Yep. Okay, then thank you for those answers. So very quick reminder to our audience members, we just have a couple of questions left now. So if you do plan on sending in any questions, please do so now before we finish so that we can catch them in time before we finish. But if you do want to send in any questions later, that's fine. You still can, even if you're watching this webinar on demand. We can send your questions in, and I'll forward them to our speakers here today. But more on that later. For now, our next question is to Uwe, which says, are there any other unit operations within a continuous manufacturing process your systems can be applied to?
Yes, I would say yes, but that's I would say granulation processes are one area where it really makes sense to look into. Maybe also things like doing ID on feeders, so like an in-process identification in the feeders is something which is feasible to our opinion in a contained environment in that case. And yeah, we haven't really looked too much into coders. We have had applications in coders, but they're kind of challenging because of the high specificity of the equipment every time. So we think that NIR can do quite a good job in a couple of cases there, but for now it's not our priority to look in that.
Okay, thank you. And Richard, anything you'd like to add here?
No, spot on.
Okay. Right.
Just
good then. Okay then. All right. So our next question to Richard then says, with regards to total tablet inspection, are you able to get a percentage label claim calculation for each tablet? If so, do you have any thoughts on what's the appropriate S value to use for the USP 905 calculation would be?
So I think that's really an assessment necessary to do on a case by case. So there's no general answer possible, in my opinion. I showed you the OC line calculations, and we are always happy to advise and help customers with this kind of assessment to give a clear feedback on the case. It's case by case dependent, product dependent.
Okay. Great, thank you then, Richard.
And whoever asks this question can happy to come back to me to investigate further here. Yeah.
Yeah. Great. Okay then. Well, before we move on, Uwe, is there any additional comments here you'd like to share?
No, thank you.
No. Okay then. It looks like that was our last question for now. I'll keep this window open just in case any more come up at the last minute. But thank you everybody who sent in their questions and joined us here today for this webinar. You can watch this webinar on demand. Perhaps you joined late. If so, you can catch up with the start of this webinar on demand and replay this on our website by going to biopharmawebinars.com/webinars, or just join again with the same link you used today to watch the replay. And if you have any questions, any further questions you'd like to have replies from Richard or Uwe from, then please feel free to send in your questions to me or direct to them by email if they provided their email in their slide decks. And my email address is marcus.osei@biopharmawebinars.com. Okay, and before we finish, I'd just like to go around and ask our presenters if there's any closing takeaway remarks. So, Richard, over to you first.
Yeah, so many thanks for setting up this webinar. It was a great pleasure. I always like to have experts on the panel like Uwe. I think this was a great opportunity to give a wide window of what's going on out there and what is the state of PET in today's continuous manufacturing.
Well said. Thank you, Richard. And Uwe, over to you.
Yeah, I hope that together we were able to create some interesting stuff for people out there interested in PAT and continuous manufacturing. And if that's the case, then it was good.
Very good. And I would also like to thank our presenters for joining us today. So once again, that was Richard Steiner, Senior Manager, Business Strategy at Pharmatech Associates, a USP company. So Richard, thank you for joining me today. And last but not least, Uwe Kirschner, who is Managing Director at Centronic. So Uwe, thank you as well for joining me again today. It's a pleasure to have the both of you here to share your knowledge, your expertise with us. So thank you. And we can end it here. So enjoy the rest of your day, everyone. Okay.
Take care.
Thank you. Take care. Bye. Thank you.
Bye-bye.